Yep.
All right, and here we are, everybody. Welcome to our live stream series that we're calling Meet a Scientist. This is a weekly live stream session that we run from here at the Albertson Research Center at the St. Boniface Hospital in Winnipeg, Manitoba, Treaty 1 territory, the traditional territory of Anishinaabe, Cree, Ojibwe, Dakota, and Dene peoples, and the homeland of the Métis Nation. So in this weekly live stream, really we're trying to have conversations with people from around our building here and around the research campus here at St. Boniface Hospital. And really the job is to try and find a little bit more out about what work goes on around here, but most importantly to meet some of the people who do the work that goes on here and kind of learn about their own passion for the things that they do. Today's guest is a special guest. He's someone that I've known for a very long time, even before he ended up here at St. Boniface, is Dr. Mohammad Sabir. We just all call him Sabbir around here. That's how I first met him too. He always has so much to share. I always love chatting with Sabbir. He's always got interesting things to share, and he'll have a bunch of interesting things to share with us here this afternoon. So he came to me with, sometimes we say, oh, we don't want these to be big research presentations. We really want to learn about you. And Sabbir came to us with an idea like, he really wanted to talk about what he called the yin yang of science and art. And this is a perfect topic, and it's something I think Sabbir is perfect to explain to us. I think, if you're watching this right now, you've probably seen this symbol in front of you before. Maybe you've heard this concept of yin and yang. This is from ancient Chinese philosophy, but it also comes up in literature and philosophy from around the world. This idea of duality, of having two opposite things that somehow come together to form a whole, right? So often these ideas that we think of as different, like science and art, they somehow come together and complement each other. Am I right on that, Sabbir? Or is that how you see it?
Absolutely. I mean, I was not sure if I'm online or can people seeing me right now?
Yeah, you can see you right now.
Great.
There in the corner. Yep.
So first of all, hello to everybody. And I'm glad for your nice introduction. It's really impressive. Yes, I mean, I do agree with you. Like, we all have our specific specialization in a particular field, but we also have passion in other forms of cultural thing. For example, art, visual art, or music, or things. And
yeah, and you're someone I've known for a long time who really does bring those two things together in a lot of the work that you do in the lab and outside of the lab as well. I see that in you all the time, and we've had a lot of chance to share these things over time. You work here. You're with you're in CCARM, the Canadian Centre for Agri-Food Research in Health and Medicine, and you work hand in hand with Dr. Taylor and Dr. Zaradka, who've been on this chat series before. But I know your path has taken you, through many different things. When I first met you, you were working at Cancer Care Manitoba in Mike Mowatt's lab. Can you tell our viewers, anyone who's watching today, about kind of what your path has been to get to where you are right now? I know it looks like you're sitting on a sunny beach somewhere, but
not physically there, but mentally there, of course. So yeah, I have a long journey. I crossed Atlantic, Pacific Oceans and came here from India. That was almost like 10 years ago. I joined as a postdoc in Dr. Michael Moore's lab at Cancer Care. And that's a place where I met your wife. I mean, we've been working at the same lab at that time. And so I came here with a postdoc fellowship and joined Dr. Michael Moore's lab to kind of continue what I have been doing in India, which was I was studying cancer and trying to understand the molecular factors which were associated with cancer. So basically in India, what I have been doing is studying head and neck cancer and map the chromosomes to find out a deleted region, because chromosome contains all our genetic material. And yeah, during cancer, some of these genetic material are changed due to mutation, and that makes the cell go completely in an uncontrolled manner. It just continue to divide, and then it becomes a tumor. And when that tumors acquire more properties for more survival and other factors, then it start moving to other parts of the body and then creates another tumor. So that's when it is called metastatic tumor, and that's basically cancer.
So
So my job was
you started there in cancer. That was kind of your first big. Was that as a postdoc or as a
that was my PhD project. So I
project?
was actually yes. So that was my PhD, and during that PhD, I was working with head and neck cancer. So I used to collect tissue sample from surgical leftover tissue from cancer patient, and then I was trying to analyze the chromosome 3 and 13. So those were two chromosomes I've been given as my project, and I've been studying the region of those chromosomes in those cancer tissues, which part was deleted. So the idea was that if we find a common deletion region in different cancer, then there might be some gene which is responsible for cancer. So we were trying to identify those genes are called tumor suppressor gene, and so we are scanning the different chromosomes to identify a small segment of the chromosome which gets deleted in different cancer. And but anyway, so that's how I finished my PhD on that work.
Yeah, and now you're in now in our in CCARM here, who's we don't study cancer in CCARM necessarily, but it's always neat to see how the things that we start out learning kind of take us on a path. I'll just share with our viewers really quickly again from here. Sabbir, you sent me a couple of the things that you have learned over time. We're not going to go big into these, but I mean, just to show you, Sabbir has this wonderful path from cancer all the way to what you're studying in cells now, where you're talking about different signaling pathways and things like this. We don't have to get all into that. But what I wanted to highlight from these pictures before you share some of your art with you is, along the lines of our discussion today about science and art, I flipped through these publications that you sent me earlier that I looked at, and just some of the pictures from these, even if you don't understand what it means, and I look at this and I hardly understand what it means either, even if you don't understand what the figures represent, there is some art to it, isn't there? There's always some art to presenting the things that we learn, to presenting sharing the knowledge that we've gained in our experiments and in the things that we're doing in our labs. There's a big part, art is involved. I look at this one and I think, these are things that I could hang on my wall, right?
Yes. So these are actually electron microscopic image of mitochondria, which is a tiny machine inside the cell that manufacture all the energy for all our bodily function. Yes.
And I think a lot of our viewers, you ran that past pretty fast, but I think a lot of our viewers would have heard of the mitochondria, right? Like even if you're in grade 8, you've probably heard of the mitochondria, as the powerhouse of the cell. And these are actual images that you can take with an electron microscope of those organelles inside those cells. Is that what we're looking at?
Yes, absolutely. So these tiny ball-shaped structures are mitochondria. They are slightly distorted because they were isolated from inside of the cell. But inside the cell, they look pretty much similar. And then we can study their function, and we can study what proteins are working within that cell compartments and all sorts of things, which is very exciting.
Yeah, and I've always, as long as I've known you, you've always done your art comes in with your science, and I've always liked I don't know if people all realize this, but any microscope now these days, like it has a camera on it, so you can take a picture of anything you're looking at. You can snap a picture with your camera, whether it's a super crazy electron microscope or whether it's a light microscope. We can take pictures of these things to see things that you can't see with your eye, right?
A lot of thing in biology is actually visual. That means you are observing something, you are recording something, and then you are coming to a conclusion. Of course, we cannot see molecule, but we can see bigger macromolecular structures. And that's why, I mean, photography or understanding the imaging process and how you can represent those images to to appeal or to make your make science more understandable. And that actually in that particular area, my experience with photography and different forms of visual arts helps me a lot. And I try to integrate both. Like I try to see how I can implement those knowledge of photography or photographic skill set into science to make my scientific data more presentable or more understandable.
Yeah, and
And
I like what you said there. It's really a way of making our world more understandable. And I think people, there's people who are really good at taking photographs out in nature and taking photographs of big things. But there's also
yes,
people who are really good at taking photographs of small things that we see in science too. I was just going to point out over this shoulder here. Here's a picture right behind me that you gave to me a number of years ago. It normally hangs above my desk over there, but I brought it in. Of the solar eclipse. When was that? In 2017.
yes.
You're good at taking pictures of the big macro things, but also the pictures of the big micro, of the little micro things too. And there is an art to it, isn't there?
So that was the time when the solar eclipse happened across the America. So I actually drove to America, like in Jasper, on the top of a mountain, and then I spent overnight to capture that image. And it was a wonderful experience. And I started from here, from St. Boniface Hospital. Like I literally shut down my experiments and jumped in a car and drove there. But that was the thing. Like it's not only about biology. Like biology is a multidisciplinary science where you can integrate chemistry, physics, all sorts of things, and you always find a link between different streams of basic science, and then you can integrate that. So a little bit knowledge of physics and chemistry really helps biology keep moving, or you being successful in biology.
Sure.
And I always try to, that was the motivation, like to understand these astrophysical objects. And then here we are working with biology, living organisms, and we are looking at tiny molecules and trying to understand their function.
Cool. And I know
Yeah,
you have, I think you've brought along some images and stuff that maybe inspire you or interest you. And this is one of the things we love to do in these chats is one of the questions I always ask is, how creativity is involved in your work. And I know you've brought along some images that you think show some of that art of science and show some of the beauty of what we can see through many different lenses, right? Whether it's lenses that make things smaller or lenses that make things bigger. I think you got some things to share with us. So please go ahead. What are we looking at here, Sabbir?
okay, I give a brief introduction. Like this is kind of a continuation where we are discussing that what I did during my PhD in India. So in India, like when I was trying to map those common deleted region on the chromosomes, then I kind of thought that the next step in my profession will be to delete those genes in an organism and then try to understand that what happens. So that is how we study a lot of genes function. So one way is to delete a gene in a mouse and then look at the phenotypes, like whether the mouse is developing cancer or some other disease. So this is a picture showing two mouse embryo. So on your right is actually a placenta. So this circular object is the placenta.
Okay.
And then this is the mouse embryo, and you can see the head. Can you see actually my mouse moving?
Yeah, yeah, we can see it.
This is the head, and this is the spinal cord, and this is the heart and the liver, and you can see the tiny limbs which are forming. So this is a very early stage embryo, and this green color is representing that the expression of a gene. So like in human being, we don't know exact number of the genes, but it is estimated somewhere 30 to 40,000 genes or something like that, and each gene has a specific function. And one of the way to understand these genes, and of course we cannot delete a gene in human because that will be unethical, but we can definitely delete a gene in a mouse because they have a lot of common physiology and anatomy structures. And that's what I did here. So I was working with a gene called deleted in liver cancer 1, and this particular gene was found associated with liver cancer. So when I joined to Cancer Care Manitoba, Dr. Michael Moore's lab, we formed this gene, and then we tagged a reporter gene because a gene forms a protein, and the proteins are kind of transparent molecules. But if you want to see a protein, you tag it with a fluorescent protein or sometimes enzyme, which can give you a color by catalytic, by a reaction, chemical reaction. So here we tagged the gene deleted in liver cancer, or I'll simply say DLC1. We tag the gene with another enzyme that produces a blue color when you scan the embryo with certain dye. So this is exactly, the blue color is forming exactly the region of the tissues where the gene is expressed. And this is abnormal. I mean, you can clearly see that this is kind of abnormal because the placenta is very small and the embryo is also very small with abnormal head structure. And you can see a very enlarged heart. So in this embryo, both copy of the gene was deleted, and that's why this abnormality was formed. So that's how we understand that this gene had some role in embryonic development.
Right, and I think that for a lot of people watching too, the idea is like, you know, if you compare a healthy cell and a cancer cell, and we look at those two cells in many different ways, when we say genes, sometimes, you know, in the healthy cell, some genes will be working properly, or in the healthy cell, all the genes should be working properly. In that unhealthy cell, some of those genes might not be working properly, or they might not be on at all, or the wrong ones might be working. And this is a beautiful way of actually visualizing. You can actually see if that gene is working and where it's working. You can actually see where it's working and compare that healthy and sick, right? And that's really what it comes down to, whatever disease we're studying is, let's compare the healthy things and the sick things. Yes. And figure out what's different. And here's a beautiful way of representing that for us.
Absolutely. So this was, and this is I mean, so my creativity, my photographic sense is how I lighted this embryo to capture this translucent image so you can see the color. So this is where you merge your artistic sense or artistic skill with imaging, like, you know, scientific object. But anyway, it's very interesting, this concept of gene. Like, I think I actually have some small demo for the kids who are watching. I'll come to that, but I want to show you some of my images, microscopic images, which might be interesting. So this image, we just talked about chromosome, right? I mean, this is a image of chromosomes. So the chromosomes, these tiny rod-shaped structures are chromosome. And here you can see that there are different number of chromosomes. In human, we actually have 46 chromosomes, and they are like 23 pairs. So 23 chromosomes we get from our father, 23 from mother. During fertilization, they come together and they form the single cells, and then we all develop from a single cell embryo, which gradually differentiate. So anyway, it's a beautiful picture, and it's kind of false colored because we put different colors to visualize molecular structures. And here,
I was just going to point that out too. Is it looks like a little bowl of spaghetti or something.
Exactly.
But when you look at that little scale bar, I don't know if our viewers have noticed that down in the corner where it says 10, and it looks like a little U. That means 10 micrometers. That tells us how small this bowl of spaghetti is, right?
Yeah. From here to here, it is 10 micron, and 1 micron is like 1,000th of a millimeter. So you take a millimeter scale.
That millimeter on their ruler, it's like taking that millimeter and dividing it into 100 pieces, right? Like
so in each of our body cells, there are 46 this kind of rod-shaped structure. And you just mentioned one thing, spaghetti. So I already got one. So this is like one DNA molecule. And you know, like so if you connect all the DNA 46 molecule, that will probably roughly about 173 centimeter or something like that. And the gene, I'm just putting a red colored dot here. So this is like a gene. So each chromosome or a DNA molecule has large number of genes along the length of the molecule. So what happened here, like, so we have like 46 of this kind of string. And then if you combine all of them together, make a ball and then put it in a small container, then this is your nucleus. And this whole nucleus is going inside a cell, which is let's say this is your cell. So this is a cell with a nucleus inside with 46 tiny threads, simply spaghetti, right? You know,
And from that one little package and that one starting cell, right? That one
exactly. So this is
fertilized egg creates everything else. Yeah.
one single cell. Now, the photographs which I'm going to show you has something to do with the concept of this DNA molecule. Now, the problem with DNA molecule is that they are very long and thin thread. It can break anytime, and that can cause damage to the cell or bring abnormalities to the chromosome. But what happens during cell division, when a cell divides and form two cells, then you also have to replicate these chromosomes. You have to replicate these chromosomes and equally distribute into two cells. And that's a huge problem. Think about that. You have an entangled mass of threads, and your job is to separate them equally. That means first you have to make from 46, you have to make 92, and then these 92 chromosomes or DNA molecule you have to separate exactly into 46 and send them to two cells. So one way the cells are really very clever. I mean, of course, we human beings are intelligent creature, and our cells no doubt have a lot of intelligence. I mean, of course, it's not literally intelligence, but anyway. So one way the DNA solved this problem, like how to separate 92 strings of DNA molecule into equal. One of the things is that if you take a thread and just start coiling it, then it becomes shorter and shorter. So you just start coiling, and then it gets shorter and thicker. And that's what happened in this chromosome. So that's why each chromosome is a rod-shaped structure, because this is a highly coiled chromatin molecule, which is basically a very long molecule. Sure. Now, I'm going to show you another picture. This is also very interesting, because here you can see that there are two cells, and the cells were actually broken down because of the preparation, the way I made it to visualize this chromosome. But you can see clearly, like, this is one chromosome and there are two chromatids. So this is a dividing cell called metaphase, where chromosomes are replicated. So each chromosome is replicated into two, and they are still held together at a point of contact, which is called centromere. And eventually they'll separate. But this whole thing is kind of decoiled, and this is the resting nucleus. So you see, it's that ball. It's that small ball with strings.
I'll bring that in for, like, you know, if anyone's watching who's in grade 9, maybe that's where they first start learning about mitosis, right? And this is what we're actually looking at right here, is you can actually capture a picture, an image of cells doing these things. So they've learned all their phases and they can probably draw it out. But this is really what it looks like, right? When it happens in front of your eyes.
And then, so there is a lot of science how I prepared this image, but I'm not going into that today. Maybe some other time. Sure. But this is a very beautiful example where you can see a nucleus with decoiled chromatin, and then a condensed chromosomes separated, and it's still whole. But anyway, this is, I found
very beautiful.
Yes, thank you. Now, next image I'm going to show you. So this is two cells which are dividing. So now I told you that these chromosomes, this rod-shaped structure, has to equally distribute, and this is what's happening. You can see the this cell. So there is a structure. This is actually called anaphase, which is so if you divide the metaphase, which is a cell division process of normal somatic cells, then the first stage is prophase, then metaphase, which you saw previously, where you see the chromatids. And then the third step is anaphase, where the chromosomes segregate equally. And this is what's happening. So during segregation, the two poles of the cell, they form some kind of strings, and they connect with each chromosomes, and then they pull them towards each part. And then that's how they segregate equally. And you can see the chromosomes are arranged here, and they are pulled apart by these fibers. This is actually a protein.
So in this one, just to make it clear, like in this one, the blue is the actual chromosomes?
Yes. So we kind of
a special staining technique where you can actually color different parts of the cell, or different molecules in the cell, color them in a very specific way, right?
Right. So here I colored the chromosomes with a different color, and then the protein which is pulling the chromosomes towards the poles with another color. So that's how I can differentiate. But these colors are kind of false. They're not literally blue. It can be any color, but they have a different wavelength of light was used to capture them. And then you can false color based on your artistic sense. I mean, somebody may want to paint it pink, which is fine.
Yeah.
But that's a different thing.
But I think it's the same. It's the same as when when kids out there, where you take a picture on your phone and you might want to touch it up later so things look a little
Exactly.
different or things pop in different ways or whatever. And I always say, the idea in photography is good photos. Um, Good photos make you think about something, right? Like make you consider the circumstances or something bigger beyond just the image. And I think this says to me, when I look at things like this, like, and you touched on it earlier before, is how coordinated this really is, and how it's so complicated to do this, right? To copy the DNA, to pull those chromosomes apart and do it perfectly every time. And this always makes me think, even in the time we've been sitting here for 20 minutes, how many times has this happened inside your body, your cells replicating and going through these divisions, and that it happens most of the time, it happens perfectly, right? Like your
Perfect.
body, your cells can do
Without any mistake. Yes,
right.
without a single mistake. It's happening most of the time.
Yeah.
And that's the beauty of science. Like, like you see this thing and then you realize that this same thing is happening in all over your body. Like your blood cells are dividing, your bones are forming, right? The
Yeah.
skin cells are growing. But again, like as I mentioned, like again, you integrate your artistic sense with your scientific skill to make this image more appealing. And also, like, you have to spend a lot of time behind the microscope to capture two cells which are perfectly one and
Sure.
in a stage of anaphase, and then another cell. So these two cells on the left, they are more separated, and you can still see they have a connection which is maintained. And then the chromosomes are perfectly separated into two cells. So these are two daughter cells, but they're still connected because the process is still going on. So you can see some leftover tubulin fibers, which are called spindle. So these spindles are still attached to the cell. So I kind of spend a lot of time to get the perfect. So this is my concept of photography. Like, it's more like framing, how you frame the picture, what you want to include in that picture, what is necessary and what is unnecessary, like, distracting.
Yeah.
So I felt good, like when I found this visual field which capture one anaphase and one telophase.
Yeah, there's patience involved actually, and this has got me thinking actually. Dr. Ian Dixon, who works here too, does beautiful astrophotography. So he does galaxy nebulae and galaxy pictures. But there's patience involved. I mean, whether you're looking through a microscope or through a telescope, there is a great deal of patience involved to get the shot just right, isn't there?
Absolutely. It's a lot of waiting. It's a lot of waiting to get the perfect picture. And then I want to show you this picture. Now, I always like this picture because it kind of captures the variation in the shape and size of cells. So different cells have different function, and they also have different shape and size based on their function. So here you can see a cell which is nice and flat, and this cyan color inside the cell is the nucleus. And then the cell has a lot of extended process, which is cytoskeleton. And
Right.
this
Almost like a fried egg, almost like looking at a fried egg from the top, isn't it? Like
yeah, or like a tent, like a tent with multiple poles, and
sure.
that is maintaining the shape of the cell. And then this is another cell, which is more like a fibroblast. And you can see, and this is a tiny cell, which is some kind of blood lineage cell, which is really tiny. So it's not necessarily all cells have the same shape and size. So this picture captures the different
Yeah,
variation
beautiful. Beautiful.
within the cell shape and size. So I always like this picture. And the next image, I want to show you the same thing. So again, like here you can see that inside the cell, now the nucleus is hollow here. I did not scan the nucleus, but the cytoskeleton, which is made up of different protein fibers, they hold the structure of the cell, and they give the integrity to the cell. They maintain the cell shape, and they also control cells migration, mobility, because a lot of cells in our body are constantly moving. And the cytoskeleton helps them. And a lot of different protein molecules, they form some kind of filaments, microfilaments, and different types of filaments. And here you can see those filamentous structure. So this whole thing is a cell, and then these are the filamentous structure within the cell holding its three-dimensional structure. And you can see how intricate it is. And also this green color is some kind of vesicles. And the cells have all sorts of function. They're ingesting food, and they're also excreting materials like a byproduct of metabolism, and they spit it out. So these kind of recycles are inside. They're constantly interacting or engaging with their outside environment, be it your blood or even like air. Like for example, the epithelial cells in our lung, they're constantly breathing oxygen.
Well, and I think what pictures like this always reveal to me too is, when we're in grade 8 and we're labeling that cell diagram for the first time, right? You label your cell and you have a couple of mitochondria and you have a little ER over here or whatever. And then you just picture it all being this empty space of cytoplasm, right? But it's not really empty, is it? Like the wonder of the microscopic photography is that you can show us how much is in there. Like it's jam packed with stuff, all these fibers and proteins and all these things going on. It really reveals the reality behind those images those diagrams we label, right?
Yes, and also a limitation in seeing things, like it is a gel-like substance. Yeah. Cytoplasm is like a gel, but within that gel, when you start seeing this different molecular architecture, then you see more things, you see more and more and more. So that is the beauty of science, like how the deeper you go, you discover new things. So when I see cell, I feel myself like a pioneer, like I'm landing in a alien space and kind of observing things which nobody has ever seen before. So that is exciting for me. And that drives my science. Like every day I discover new things, and that keeps me moving, at least not boring. Like, like I'm not doing the same thing daily basis. But anyway, I want to show you another picture. So this is another cell. It's a cancer cell. But in this image, I always think that cells may have some mathematical knowledge, because you can see that there are some fibers which are perfectly triangle. This triangular appearance of the cells is just fascinating. Like how do the molecules know that they have to organize in such a perfectly triangular shape? It's like a beautifully designed architectural structure inside the cell. So there is a branch of science, like they actually draw ideas by engineers are studying I think it's bioengineering, or even the architecture, like architectural inspiration that comes from studying biological objects. So this is a beautiful example. Like I was fascinated by this triangular appearance of the fibers. These are again, these fibers are called stress fiber, and they are made up of some kind of actin protein. And these are anchoring points. So the green color is actually another protein called vinculin, and these proteins are anchorage point on the medium on which the cells are growing. So each fiber is connected to this anchorage point, or anchored on the medium. And then the nucleus, you can see the blue color, and rest of the cells are cytoplasm. So this is a beautiful example. But as I previously mentioned, that we always put artificial color. It's not really green color, but we can put another color. For example, here, I just colored them differently. I put red color for the fibers. And so this is just to give the idea of the audience, just to see how we false color these images. But they were captured at different wavelengths of light to image different proteins, and then they were merged together, and then they were artificially colored. So this is again the same cell with a different color.
Almost as if if kids can picture the idea of, if you had a camera that could take a picture in visible light, like regular, you have a picture that like this, but if you had a camera that could also take infrared images, you could get a different image and kind of stack those on top of one another. And that's kind of what you've done here, is look for these different wavelengths of light coming off of these cells, and you can kind of stack those images on top of one another.
Yeah, exactly. Something similar we use a filter, like optical filter, which cuts off the lights. It's like your sunglass, cutting off one particular wavelength of light and allowing others to let in. So we apply the similar technology.
Yeah, very nice.
Yeah, thank you. So next cell. So this is actually, so some of my research, I did a lot of different areas of research. I have experience in neuroscience research. Now I'm doing a lot, doing atherosclerosis and other cardiovascular diseases. So this is an image of neurons. So there are 4 nerve cells growing. And the fascinating, and these are cultured neurons. These are not like within a living organism. It's like I have extracted some neuronal cells and let them grow on a petri dish. And the neurons, one thing funny, not funny, very interesting about neurons is that they are the longest cell in our body. They are the
Yeah.
longest cell. Like one neuron cell can extend from your brain to the tip of your to your toe. So it's such a long structure. And the neurons, they are terminally differentiated cells. So that's why if they damage, they cannot grow. You cannot grow it back. And that's why a lot of neuronal damage or brain damage is kind of permanent. But anyway, here you can see the neuron cells, they're sending these tentacle-like structures, which are called axons, and they transmit neuronal transmission. They transmit signals. So, if, for example, touch or any sort of perception, and all this is similar. So I find this is very interesting, like balanced. There are 4 neurons, and they're sending out branches, axons, and which is very nice.
Yeah, beautiful.
Okay, so you let me know when it should stop because I have so many images to show.
No, I know we could go on and on and on. And you know what? We could always do another one of these and come back. Like there's so much in these that remind me of so many things. I think we could talk for hours about this. Why don't you show us a couple more of your favorites and we can wrap it up in a few minutes.
Yeah, maybe. So this is another cell. This is more like astrocyte with some extended processes. Maybe I can just go on quickly a little bit. And
yeah.
This is another cell where you can see like chromosomes. The red color is chromosome, and these are the fibers which are pulling chromosomes. But this is the tumor cell. And again, like this is the, I'm putting different color to increase this artistic impression. And again, this is a cell showing anaphase where chromosomes are segregating. Now this is an abnormal cell, so sometimes these divisions can go wrong. And this is a beautiful example where you can see that instead of 2 poles, now the cell has 3 poles, and the number of chromosomes is too much. And so this is an abnormal cell where the chromosome number is much more than it's a polyploid cell. That means the ploidy is the level of chromosome, like a normal cell is diploid, but it is definitely more than diploid chromosomes. And this is a tumor cell. And again, this is another cell which is showing segregation of chromosome. The blue color is the chromosomes and the white is the poles, which are pulling the chromosome. And you can see that it's a late anaphase where the chromosomes are already separated at the beginning of telophase. And then this is another cell where you can see that a single cell divided into 3. This is a tumor, abnormal condition, and they are still connected together by the spindle, which will be disappeared after some time. But I find it's like a grapes, like, like 3 different cells connected. So it's kind of, I found this visually very appealing. And then again, the same thing. So here the cells are completely separated, but they are still connected by spindle mechanism. And so this is the nucleus. So ultimately, eventually,
I should say there too, Sabbir. Like it's interesting when you look at those. They're pretty pictures, like they're absolutely beautiful, but that teaches us something too, right? Like it all comes back to trying to learn about, how these cells how they behave normally, and then how they change when disease happens. And we have so many different techniques that we use in our labs here. Not all of them show us the beautiful picture, right? Some of them you really have to puzzle through all these things. But sometimes just the image really can bring it home to you, can it?
Yes. Yeah,
what are we looking at here?
Oh, this is like this is a cell where I have stamped only the nucleus. So you can see the normally nucleus are very round structure, and they are not homogeneous, but they have a lot of structures visible. Maybe I'll talk some other day about these subnuclear structures. I have a whole series of images. And this is another cell where you can see the chromosomes are segregating. So I also love this combination of colors and the appeal of the image, like where you can see the chromosomes separating. And again, like this is again abnormal cells with abnormal spindle structures. And again, this is the anaphase cell, and chromosomes are segregating. Now I better go. I better. It's becoming more monotonous, right? So I kind of show some of my photographic skill. So this is an image I took in Oak Hammock Marsh. When the weather is good, I just explore the nature. And this is sandpipers. They're flying, and I kind of captured at a particular moment of these flying objects when they're all perfectly in a single plane. So you can see these white strips on their back. So I found this image very fascinating, and I can capture the motion here. So So I thought maybe I show you some of the photographs just to break the monotony, because I have been
Yeah,
talking about science a long time.
yeah, sure. I share some of these with us. I think that the neat part always is, there's patterns, isn't there? And even in all those pictures of the cells you showed us, there's certain patterns that jump out at you. And as a good scientist, you always have to be looking for the patterns, and you always have to be looking for where the pattern changes or where the pattern isn't repeated properly. And even going back to your talk about cancer cells is we're always looking for that. Oh, it should be this proper, telophase, anaphase, all your different stages. But
Yes.
sometimes those patterns, the things that we should see shift. And even when we move to nature photography, like you look at this picture in front of us right now, there's patterns here too. Like, why are all those birds angled in a certain way? And why are, there's so much to observe, whether it's through the microscope or whether it's out at Okemos Marsh, or whether you're in Wyoming or whatever, taking pictures of the eclipse. There's these things that have fascinated people forever, right? And the deeper you go or the further out you go, those patterns just keep revealing themselves over time. And that's what jumps out at me when I look at pictures like this too.
No, absolutely. Biology is the observational science. I mean, your observation is skill. Like sometimes I discover new things just by finding out that deviation in the pattern. Like, like you expect a pattern to be observed, and then you see a deviation from that pattern, and your eye has to be trained to find out this deviation or discrepancy,
Mhm.
and then chase that. You try to understand why it happened. Is it happening every time, or is it just a anomaly or random thing? And that's why like my power of observation helped me a lot to understand biology and to discover, because I'm very good observant, and also that is reflected in my photograph. I show you another photograph. So this is a picture I took in Alaska, where you can see grizzly is kind of
I hope you
shaving
had
his.
a nice long telephoto lens for this one. Big long lens. Yeah.
It was a telephoto. Of course, I'll be dared to approach this kind of dangerous animal. And this is a beautiful picture I always cherish. This is called the super moon, and it was again the Oak Hammock Marsh. So I kind of took a so that happened a long time ago. I forgot exact year, but probably 4 or 5 years ago, and the moon was really very big. So I went to the Hammock Marsh and then set out my camera to capture the moon, and you can see the so much details on the moon and the color and the whole experience. And it was sometime in the fall, because you can see the lot of birds flying in the marsh. And this is a picture somewhere in the prairie, where I see the pattern of the clouds and the reflection and the crops, the color of canola, probably, yes.
Yeah.
And then this one, I like to, there is a famous painting called Scream. So I took it in the Oak Hammock Marsh, and this is a coot, probably scared of me and running away. But I captured the reflection of light, and it's, I mean, I feel like it's kind of screaming, like the whole image is kind of a fear, like it's running away, and this reflection of light, and it's literally walking on the water. So,
Beautiful.
yeah, so it just and also the color, the inspiration of color comes from nature. You see the butterfly. This is the Manitoban butterfly. I forgot the name, but it's actually leaking salt from the sand. So that's why, I mean, a lot of us think that what it is doing on a sandy beach, but it's actually leaking salts from
Huh.
the tiny sand particles, and that's where you just spread the wing and you can get the perfect color, beauty of the wings. This is another butterfly. So I do a lot of photography. When I'm not doing science in the lab, then I go and explore nature, and I take pictures and I observe nature, and then I get refreshed, and then I come back to my lab, and I observe cells and
Yeah.
molecules.
Well,
That's
and
pretty much my thing.
that's the beauty of it too. I mean, it's observing nature, whether you're doing it in the lab through a microscope, like we're still trying to figure out how nature works. And there's a beauty, whether, like I say, a beauty whether you're looking through the microscope or whether you're looking through your camera lens.
Yes.
There's beauty to be found everywhere, and there's patterns to be found anywhere. And the goal in our labs is, maybe those patterns can teach us something about our health or about how we work and about our place in the universe, right? Like
Yes.
I think there's so much to be learned through the lens, and I really, really thank you for sharing everything today. Like I think what you've shown us, Sabir, and this is what we really try and show people as we do these chats is, there's science isn't just about, learning the facts in school and learning the facts out of books and things like that. It's really about how do you bring your passion for something and your interest, your genuine interest. But there's so much technology involved. Like it's this perfect mix of creativity and passion and technology and patience too, right? The patience to be able to search and to look and to find the things that you're interested in.
And I think also the curiosity. I mean, you have to have a childlike curiosity when you are approaching science.
Yeah.
Because curiosity, I mean, and no question is dumb question. I mean, a lot of students, they are kind of sometimes feel shy, like they feel it's embarrassing to ask a question which may not make sense. But I think it's very important you ask questions, always challenge the existing concept, and
Exactly.
and try to think deeper and try to interpret whatever you are seeing and observing in nature and also in your life. But anyway, I just want to show you a couple of images, just flying colors, I'd say.
Beautiful.
Butterfly, lots of butterfly, and lots of nature, like that really refreshes me. And I kind of observe the lines, how they're perfectly aligned, these
Yeah.
ducklings. And also, so these are two butterfly, they're mating. It's beautiful shape, and also these dynamics of flying. It's a lot of nature photography I do, the dynamics of nature, the shapes and lines and the colors. So this picture, I'll show you two pictures, and then probably we can discuss or end the session.
Sure.
So this is a picture where I apply my sense of color. So this is an electron microscopic section image of a nerve fiber. So we have nerve fibers that are coming out of the brain and supplying to different organs. And literally these are wires, like they're carrying neuronal transmission. And if you cut a section, then you can see that these are myelinated nerve fiber. So it's a thick myelin sheath, which is like a layer, insulating layer around the nerves, so that two nerves do not, make contacts with each other, or
Yep.
their transmission do not get connected. And then these tiny dots are actually mitochondria inside the neurons. And so this image was taken, and then I put different colors. So I thought it will be a nice wall photo. And if I just put some color, so I filled up different color in different nerve fibers, and then made this image. And it was fun. Like I kind of found it's very relaxing. I'm just, instead of knitting or doing something like that, I just started filling up this section of a nerve fiber with different colors, and kind of it's visually appealing. And then I also do a lot of cartoons. I like cartoons. I'll probably show someday some of my cartoons, because I try to integrate cartoons into science and then some fun. So here, it's a section so the mouse is sitting on the window, and the window is kind of opening to your brain, and in the backdrop is a section of a brain. So you can see all these neuronal process. So this yellow color is a neuronal process, and this is the mitochondria, which is a powerhouse of neurons. And each color is showing a section of a neuron on a cross section. So basically, it's a mouse brain, a tiny mouse looking through a window to its own brain. So that makes complete sense, right? So anyway, I found it kind of fun to put the mouse on the front, and then put it as a background out of the window. So we are constantly looking into brain to understand how brain functions, because that is a mystery to us. We really don't understand completely how we store memories, how our consciousness works, or how we learn things. I mean,
Yeah.
we understand at some level, but we need much deeper understanding to really cure disease.
Yeah, and I think this is a great image to end it on, Sabir. This is we talk about like it's perspective, right? So there's this concept of perspective that exists in art. So as we learn about art, we know perspective is a big piece of artwork, and perspective is a big piece of science too. Like how we look at things and how we train our brains to look at things and understand them. I think you shared so much of your own perspective and your own perspective through the lens. I think that's been really it's been a beautiful tour of some beautiful images. We'll definitely have you back maybe maybe talk about some of your cartoons and your other artwork later down the line.
Absolutely.
You've always had a great way of communicating. Communicating through images is one way that I think appeals to everybody, and we definitely want to thank you for joining us today. We'll definitely have you back on in the future, because obviously we have a whole lot to talk about.
Yes.
It could go on for hours and hours. So I'm just going to switch over to my screen and wrap things up, but I will thank you, Sabbir, for joining us.
Thank you very much, Steve. It was always my pleasure to meet you and to share my images and research experience, and just to talk to the audience of your beautiful Youth Bio Club members.
Yeah, and I always say, if anything rubs off on kids, whether whether it's the science itself, it's the passion. People love what they do, and people love learning new things. And I think all of us, from when we're kids ourselves, it's that you said it best. It's that curiosity that it's all about. And whether that's science or whether it's art, there's always a curiosity and a creativity involved. And I thank you very much for sharing with us this afternoon. So I'll wrap it up here for everybody. That'll end our chat today, but thanks very much again for Sabbir for joining us on our weekly Immunoscientist series. Next week, I think we're having another guest from CCARM. That's Luis is going to pop down and talk to us about what he's studying. And just make sure to subscribe to our YouTube channel here for updates. Check us out at youthbiolab.ca. Follow us on Twitter and Instagram to keep up with what we're doing and what's coming up next. So thank you, everybody. Thank you, Sabbir.